So let's get started. Good afternoon, everyone, and thank you for joining us today. It is my pleasure to introduce our speaker today, Dr. Kathryn Lin. And Dr. Kathryn Lin is a tenure-track assistant professor at the University of Washington's Department of Biostatistics. And he was formerly a post-doctoral researcher at the University of Pennsylvania's Wharton Department of Statistics and Data Science with Dr. Nancy Zhang, and also completed his Ph.D. at Carnegie Mellon University's Department of Statistics and Data Science under Dr. Kathryn Roeder and Dr. Jing Lei. His research focuses on studying cellular mechanisms from single-cell data. He develops novel methods using ideas from matrix factorization, network modeling, and changepoint detection to derive new insights for advancing single-cell biology. Personally, I met Dr. Lin in a lot of statistical conferences, and his talk is always very fantastic. So today we're very fortunate to have Dr. Lin here to discuss his latest method, eSVD-DE, a cohort-level differential expression in single-cell data while matrix factorization. So please join me in welcome Dr. Lin.
Well, yeah, thank you. That's high expectations I need to fulfill. But it's been wonderful being in this department. I had a great time so far, and hopefully I will return the favor and give you a great time for the next hour or so. So this entire talk, I'm basically a diehard single cell person now at this stage of my career. So this entire talk will be about realistically cell biology and specifically single cell RNA sequencing. And for those of us who haven't worked on this data, even though I'm sure you've heard many talks about this before, it's basically a type of technology that allows us to profile individual cells, many thousands of those cells, and we can measure the gene expression profile for each of those individual cells. So for example, this is a cartoon. You can imagine if I sequence someone's skin tissue, for each of these cells, they form a row of my matrix. And then for each of my P1 genes, typically we subset it down to just like 5,000 of the most important genes. Then we get a count matrix. So for each cell, I get a count 0 to some positive number, being like how many mRNA transcripts was in this cell for this gene. And then that gives me a sense of like how active that gene is for that particular cell. Now, this talk, as the talk title suggests, this cartoon is of one donor. And now the question is, I have many such of these donors. What can I do with this data? So yes, from one donor, I can profile his or her, in this case, skin cells, many thousands of them. But now, due to the thanks of the technology, if my pointer doesn't work, that's fine. Thanks to the technology, we can now sequence tons of people all at once. The cost of these single cell sequencing prices have come down a little bit. That makes it affordable for consortiums to profile the single cell data for many people. I'm just going to show you some papers of this. So this is, to this day, the biggest cohort that I know of. It's a blood, which is the easiest tissue to get, cellular data for. And so in this particular example, they're sequencing blood from 900 plus people, which in the single cell world is a lot. So for those of us who work on genetics, you might be wondering 900, you can't do any fancy GWAS on 900 people, but on the single cell level, this is considered a huge amount of people. Now, just to give you another example, for those of us who don't work on the immune system in the blood, one of the other data sets I'm really interested in working on nowadays is of the brain, for reasons I'll tell you at the end of the talk. And so one of the biggest donor consortiums for brain, of course, someone needs to pass away and donate their brain to the consortium, is of 388 people. And now you can get all the single cell profiles, all your neurons, astrocytes, microglia. And for each cell, you see the gene expression profile for each one of these individual cells. And this one, over 2 million nuclei, is for 388 people. So this is just to give you a sense that these cohorts are getting bigger and bigger. They probably won't break into the thousands anytime soon, but they're certainly quite computationally massive nowadays. And so the question is, okay, so let's say I want to do differential gene expression testing. And for those of us who've studied single cell or even studied genetics, you might wonder, there's probably nothing that's special about it. There's tons and tons of differential expression methods out there. And what makes it so different now that we have many people? And that's going to be the main focus of today's talk. So this slide may or may not make much sense depending on how familiar you are with single cell, but mainly my goal is to convince you what the bottleneck is, and the bottleneck is a regression issue. So usually when I profile all these genes from all these different people, I'm going to analyze if one gene is differentially expressed between my cases and controls. For example, as a very, very concrete example, let's just imagine I profile a bunch of people. Some of them come from, let's say, brain cells of individuals who were autistic. Some of them came from brain cells of people who were not autistic, so classic case control. And I'm going to ask, is this particular gene one of my genes of my 50,000 genes or 5,000 genes? Is it differentially expressed? Is the mean very different in the cases versus the ones in the controls? And so what makes single cell a little bit tricky, especially when I move to a human analysis of this large cohort, is that in general, when I think about one gene, so one column of my matrix, one of the realities I need to handle is that this gene is incredibly sparse. Now, that by itself is not the biggest challenge, but it's going to be a bigger challenge, which I tell you the other side of the equation. So just focusing on the gene expression count, just for those of us who haven't worked with single cell data before, you should think that for any particular gene, a typical gene in a typical data set, most of those values in that column are zero. So, 80% of them, 90s, I think usually it's around 70 to 90% of the column are exactly zero. Usually 15% of the counts are one, and then maybe the last 5% are even like two or higher. So this is just to give you a sense that you're basically working with almost all zero matrix, all zero vector. And what you want to control for, just like in any differential expression analysis, you might want to control for the age, the sex, the smoking status of that individual. And you'll realize this is a little bit funny because even though I'm analyzing on the single cell level, of course, all the cells from the same donor have the same covariates. So each donor contributes 500, 1,000, 2,000 cells, but all those cells essentially come from the same donor, so they have the same X covariate, the same age, the same sex status, the same smoking status for all 500, 1,000, 2,000 of those cells. So essentially what makes this tricky is that we have a very, very sparse matrix, and I want to adjust for covariates that come from the donor, but because I have so many cells per donor, every covariate feels very discrete almost. So yes, even though age is a continuous variable, if I have 38 donors and I have 38 discrete age values over like thousands and thousands of cells. So this is what makes it tricky, is that I want to adjust for these biological confounders, even though I'm working with really, really sparse data. So you can ask yourself, if I have a vector of all zeros, how can I sensibly control for sex and be sure that I am adjusting for it appropriately when my vector is basically all zeros? So in this talk, basically, I have two goals. One is to convince you that this does make a difference, and how do we solve it? So most of the single cell work from, I would say, the start of single cell, which is roughly around 2010 to, let's say, 2016, 2017, they were mainly designed for cell lines and clonal mice. So essentially systems where you did not have a lot of biological variability. And now that consortiums are now collecting massive, massive amounts of single cell data from humans, we now need to think about how to control for all these biological confounders, age, sex, smoking status, on single cell data. And that's going to throw a wrench into some methods. So that's going to be point number one. Point number two will be more statistical in flavor, because as I'm describing this problem, some of you might have already some solutions you have in mind. And one of them is going to be about imputing the data to impute away all those zeros. But then there's going to be some statistical road bump I need to solve, that imputing the data will naturally inflate the type 1 error unless I do it appropriately. I'm going to tell you a little bit about that. And that's going to be my statistical goal. And overall, though, I think this theme that imputing will inflate your type 1 error unless you do something to counteract it, I think it's a theme in many, many areas of research, much beyond single-cell RNA-seq. In the tail end of this talk, for my friends who work in genetics, I'm going to say a little bit about why this problem isn't the biggest deal in genetics specifically, because we're doing imputing of the SNPs all the time in genetics. But certainly for single cell RNA-seq, if you're analyzing metabolites, if you're analyzing protein data, certainly this theme will probably resonate. So there's going to be a slight distinction, I'll say at the end of the talk, about why genetics doesn't really suffer from the same problem that the single cell gene expression side does. But all that to say, it's always good to have a plot that visualizes what kind of data we're working with. So in a differential expression testing, as I mentioned, it's become more and more complex in recent years because now we have many, many individuals and we have strong confounding effects. After all, if I'm studying, I'm going to make up some biology. If I'm studying autism, but it's also correlated with sex, I don't want to find just genes that are correlated with sex. I want to find the mechanism of autism. So I want to control for strong confounding effects. Age, sex, smoking status certainly has an effect when I analyze single-cell data of the lung. And so the goal here is what I write is an English sentence of what a differential expression analysis is. Given a gene, is the mean expression for this gene significantly different between my case and control individuals after controlling for my observed confounders? Once again, age, sex, smoking status, things of this nature. So this plot right here, just imagine I have 30-some individuals. I'm looking at one particular gene, and this violin plot is of all accounts from this particular donor. It looks a little bit funny because we are looking at count data. That's why there's like little bumps around 0, 1, 2. And so you can kind of artificially convince yourself that the most common value by far is 0. And some of them have more non-zeros than others, but a lot of the time you're looking at exactly a value of 0. And so once again, the key points to this message is we want to test for the mean expression across individuals while controlling for confounders. And how do we do this when we have strong confounders that is, on basically vectors, gene expression profiles that are basically 0 across all my cells? So some of us who work on single cell will probably already, our gears are probably churning in our heads already. There's probably tons and tons of methods you're probably thinking of. And so I'm going to essentially walk you through throughout this talk about some of the most common methods currently being used. You might have used some of these methods yourself. So one big category of method is forget about which cells came from which donors. I just have 2 buckets of cells. One bucket is any cell that came from any case donor, and another bucket is any cell that came from any control donor. Forget about which cells came from which donors, just analyze it as case cells versus control cells. And I'm going to tell you a little bit why that may or may not be the best idea. Another one, which I'll focus a little bit more on, this is usually the gold standard nowadays for analyzing cohort data, is to construct something what we call the pseudobulk. If you haven't heard of this word before, I will describe what this is more graphically in a little bit. And then some of us might be also thinking of mixed effect models. You have a hierarchical nature, you have a donor who has a bunch of cells. Some of us who work with mixed effect models might naturally think this is a very natural plugin. And certainly there are methods that try this. Towards the end of the talk, I'll give you some empirical comparisons against some mixed effect models. But the overall broad theme that all these methods have is that they are adjusting for covariates after analyzing each gene one at a time. So for example, if I have 5,000 genes, when I apply any of these methods, it basically forgets that there's 4,999 other genes. It's just focusing on one gene and it does it in parallel. So you can kind of imagine these are methods being deployed in parallel over my 5,000 genes of interest, and then I just do a multiple testing correction at the very, very end. And so what this paper is going to do, what I'm going to tell you about, is a slightly bit different variation than any of these. So what I'm going to do is first impute all my data. I'm going to denoise it, analyzing and pulling all the information across all my genes. And then I'm going to do some donor level testing. So that's going to be the broad strategy. And I'm going to convince you that this is sane once I do a little bit of adjustment. And I'm going to tell you some roadblocks specific to this strategy in just a little bit. But to start off with, I think it's useful to just think of this hypothetical in this wacky world of single cell, that at the end of the day, what I want to convince you in this slide is that the number of cells is not the most important number. Because, for example, it's useful to think about what is different between testing for differential expression between cells and testing for differential expression between donors. And all my data here is of single cells, but it's useful to have this concept in mind. So imagine I have 2 data sets, hypothetical data sets, each with 1 million cells. So in data set A, I have literally just one case donor, one control donor, and each donor contributes 500,000 cells. 500,000 plus 500,000, I get my data set of 1 million cells. I exhaustively profile these two specific people to the ends of the earth, because half a million is a lot of cells to come from just one person. Now, in the second data set, I have 1,000 donors, 500 cases, 500 donors, and each of those 1,000 donors contributes just 1,000 cells. And so you'll notice I have much fewer number of cells per donor, but I have a lot more donors. I have 1,000 donors compared to 2 donors. So hopefully with this example, you can convince yourself that even though both data sets have literally the same number of cells, they kind of feel like they have different qualities to them. You can imagine if I profile 2 specific people to the ends of the earth, I could probably learn some very specific biology that differentiates my one case donor from my one control donor. But I probably would not expect those findings to generalize to a human population, because after all, it's just 2 people I happen to select. But if I had 1,000 donors, okay, it's not great compared to the people in this audience working on genetics. 1,000 still seems very few compared to our GWAS studies of like 10 to 100,000 people. But 1,000 is pretty good in a single cell world. You might imagine this data set, I don't get to profile each donor very accurately, but if I have 1,000 of them, if I do find anything significant, it probably does generalize much better than my data set with just 2 donors. So you can imagine there's some trade-off being done right here. So the first takeaway with this thought experiment is that to convince you that a single cell analysis of donors is different than a single cell analysis of cells, is that you might imagine data set B has a little bit less power because I just have much fewer cells per donor. But if I find anything, it probably is reflective of the human population. And data set A, even if I find a bunch of significant differences, I probably wouldn't expect that to happen if I happen to sequence 2 other donors, one case and one control. And the second takeaway in the very same vein is that what drives the power in these analyses is not really the number of cells, because if the number of cells was driving the power, both of these data sets would have the same amount of power. Power to do what? In data set B, it's power to generalize towards the human population. In data set A, it's just power to find the differences between these 2 random people I happen to sequence. It's not the number of cells that drives the power, it's probably the number of donors that drives the power. So that's another takeaway to help us wrap our head around what's about to come. So I'm going to illustrate one more cartoon to get our gears grinding, and then I'll finally tell you what my method is going to do. So now that I've convinced you that it's the number of donors that is driving the power, probably, not the number of cells, let me just show you pictorially what might happen. So in illustration A, just imagine each of these histograms represents 1 donor, and the height of the histogram represents how many cells I'm going to get from this 1 donor. You can kind of imagine in illustration A, the point is, if I don't account for which cells come from which donor, I just pull it into 2 big buckets: any cell that came from any case donor versus any cell that came from any control donor. I ignore all this hierarchical information. In this particular data set, there's 1 control donor in blue that produced a ton of cells. And so if I ignore this donor structure, I might accidentally say there's a significant difference between the cells, but I'll be oblivious to the fact that 1 donor contributed like 10 times more cells than any other donor. Now, what I'm going to tell you in just a little bit about pseudobulking, for those of us who know about pseudobulking, I'm going to reiterate this message when I talk about pseudobulking in a little bit, is that what a pseudobulk does is it collapses all the cells into just 1 vector. I'm going to add up all the cells from 1 donor, or average is probably a better word to use. I'm going to average all the cells, all the 500 cells from the single donor, and collapse it into just 1 vector. So now I have, instead of n cells in my matrix, I have n donors in my matrix now. And what I lose is information about the variance. So what you can see here, what's illustrated in point B is kind of the perils of pseudobulking, is that there could be a lot of differences in the averages between cases and controls, but what you lost information about when you construct your pseudobulk is that maybe each donor was very variable. So I'll come back to this point in just a little bit. So these are just some graphical intuitions. And so now let's actually talk about what I'm going to start analyzing. So in this particular talk, I'm gonna use one particular example from T cells and lung. It's going to be a little bit clear about why I'm going to choose this particular data set among all the data sets I could possibly choose. But this for now, just take my word for it. We're going to analyze T cells from a lung data set across, in this case, 34 individuals, 24 cases, 10 controls. I have different covariates, age, sex, smoking status that I want to control for. These, for those of us who have not worked with single-cell RNA-seq data before, this is your classical UMAP. Just think of it as a fancy visualization of my data where each point is a different cell. And what's shown here is that I'm coloring any cell from any control individual as a different shade of blue, any cell from any case individual as a different warmer shade of orange or red. And so given that this is the data, once again, my central question is I just want to find the genes that are differentially expressed between the cases and controls. But you'll realize that there's already a lot of differences. If I just look for sex, there's a lot of differences between the genes if I just look for smoking. So of course, I want to control for these things. I don't want to find a gene that's just exclusively driven by the fact that some people smoke and some people don't smoke, because in this case, I want to study the particular pulmonary fibrosis disease, not just whether someone smoked or not. And so I want to account for all of these things. And once again, as a warning, each of these genes is incredibly sparse. We're working with a count matrix where each column of the matrix, you should roughly expect 70% to 90% of the values are exactly 0. And so the first thing I'm going to talk about in a little bit more detail is pseudobulking. And this will motivate why I'm going to do what I'm going to do in my method. So pseudobulking, once again, is I start with my N cell by PG matrix. Just imagine this is my single cell matrix. All the white pixels, just to note that that count in this cartoon data set is exactly 0 at that location between that cell and gene expression. And what pseudobulking does is I average all the cells from each donor. I average all the gene expression profiles from donor 1, donor 2, or donor 3. I'm just left with a donor by gene matrix. It's a pretty simple transformation. And then if I have S donors, I'm going to do my differential expression testing on this much more dense matrix. And so the intuition behind a pseudobulk analysis is that you can imagine I'm just going to do some kind of t-test. There's obviously way fancier methods, but this is just intuition. I'm going to look for any particular gene, the difference between the average case minus the average control. I'm going to normalize by some standard deviation. And what's tricky about this is that after I pseudobulk, you imagine that because I computed the average gene expression among all the cells in a donor, and I threw out all the information about the variance within a donor, this standard deviation is miscalibrated because technically it should probably include a term about how much variance there are between different individuals and a term that says how much variation within an individual there is. But because you computed the average and you threw away information about the variance, you're going to throw out this term, and then all of a sudden your null distribution is miscalibrated because you forgot about accounting for the variation within a donor. And so this is basically my one slide intuition on why a pseudobulk method might go wrong. You're removing information about how much variability there is within one person when you do your analysis. And so this is why you might want to do a single cell level analysis. You don't want to throw away that variation because that might be useful for you telling which genes are differentially expressed. But now the question is, how do I account for that while removing for confounding effects? Because once again, the benefit of doing a pseudobulk analysis, as I showed here, is that this matrix is much, much more dense because I average over your 500, 1,000, 2,000 cells per donor. And if I don't do that averaging, I'm back to square one where I have a very, very sparse matrix. So how do I account for this? What should I do? And this is where our method is going to kick in. So essentially, it's going to be a pretty straightforward idea. First, I denoise my count matrix, and then I apply my t-test. But however, for those of us who worked with such type of strategies in their context before, this is not the best idea naively because you're going to get a massive type 1 inflation. So this slide would just be to demonstrate the problem. The next slide will be to explain why this problem might happen. So imagine I don't do any denoising. I just throw a standard differential expression method on my single cell data set. So realistically, all the genes that are highly significant, this is my volcano plot where The y-axis is the -10 p-value. So larger on this data set means that it's more significant. This should read 10, 100, I think this reads 10, 100, and 500, but the axis got cut off. But you can imagine all these genes are highly powered mainly because they're densely sequenced, and all the sparse sequence genes are sitting at no power, not because I don't know if not because they might not be differentially expressed. I just don't have enough power to identify them. And if I first just throw my count matrix into a typical imputation method, just think of a fancy version of a PCA, then what happens is all my p-values, almost all the genes start looking significant. So essentially here, these dotted lines right here in both plots are the multiple testing thresholds. And so if I smooth my genes, I remove the zeros by imputing them a little bit. So I no longer have exact zeros anymore. And then I do a differential expression test, any reasonable one. For most reasonable ones, you'll see that your p-values shoot through the roof, and almost all the genes now look differentially expressed. And certainly if you have 5,000 genes and you claim in your analysis 3,000 of them are differentially expressed, most likely something went wrong. It's improbable that literally all the genes are involved in this case, lung disease. And so why does this phenomenon happen? And so this is a pretty reproducible phenomenon. You take any single cell data set, you denoise, and then you do differential expression. You see all the p-values shoot through the roof. And I'm going to use a very, very simple cartoon to illustrate why this might happen. So I'm going to generate some cartoon data sets. Synthetically, there's going to be 6 donors, 3 cases, 3 controls. Each contributes, 50 cells. And I'm just going to literally have 2 genes, gene 1 or gene 2. And then what I'm plotting here is each individual cell. So just imagine I'm measuring just 2 genes, and each dot denotes the gene expression for this particular cell and also the donor averages. So those are the bigger dots. So let's say I do this. This is going to be a simulation. So everything is Gaussian data. It's real valued, continuous. I'm going to make my life as simple as possible. Now, if I showed you this data, there's only a 2-gene analysis, and I ask you which genes are differentially expressed. There's only 2 genes. I would venture a guess. Most of us would say that gene 1 seems differentially expressed between the reds and the blues because there's a very visual, obvious gap between the red and the blue cells. But maybe gene 2 is probably not differentially expressed. And so if you just use any p-value calculation, sensible p-value calculation on this synthetic data set, you'll see that, indeed only gene 1 is significant, gene 2 not so much. Now, what happens if, let's say, this is a simulation data set, so I can do whatever I want. Let's just say hypothetically, I denoised my gene expression matrix and then did a differential expression test. I only have 2 dimensions, so by dimension reduction, let's say I just collapse everything to 1 dimension and then do my differential expression test. So if I just, unbeknownst to me, I first project all my cells onto the first principal component, then surprisingly, or maybe not surprisingly, depending on if you've seen this before, both of my genes now look very significantly expressed. So my p-value is still 0.002 in gene 1, but now gene 2 also looks very significantly different. And the reason this happens, it's not anything that magical, is that a lot of the time in your single cell analyses, the true differential gene is also densely sequenced. So a lot of the subspace is dictated by the truly differential genes. And because you projected everything, you did a dimension reduction, you forced everything into a low dimensional space, basically that signal from the true differential gene bleeds into all the other genes by the nature that you're doing a dimension reduction. So oftentimes in a single cell analysis, the truly differential expressed genes dictate the subspace. They capture the most variance, and you projected everything onto it. So even if a gene originally looked like it was not differentially expressed, because it got projected into that subspace, it now looks like it's differentially expressed. So this is the phenomenon that explains this. So you can imagine there's a few genes here that dictate the subspace that I'm projecting onto, and I projected everything onto that. So now everything looks significant. And so this is the problem. If we do want to denoise the data to overcome the zeros, we have to account for this kind of roadblock that is stumping our way. And so how are we going to do this? So this is now 20 plus minutes into the talk. This is where now I tell you how my method works. So first, is your usual dimension reduction. There's a little bit more nuance to this dimension reduction, which I'll tell you a little bit about. It has to account for the fact I have covariates. It has to account for the fact that I have sparse count data, but nonetheless, it's a pretty standard dimension reduction for negative binomials. In step 2, this is the real trick that I'm going to use to overcome this type 1 error inflation common to single cell analyses, where I'm going to look at the posterior mean and variance based on some model and kind of adjust accordingly. And then step 3 is nothing too special. I'm just going to, with this posterior mean and variance, I'm going to form a test statistic and do some multiple test corrections. So the real novelty here is just using step 2 after step 1, if you wanted to put it in a simpler term. So let me just tell you a little bit about each of these steps. So in step 1, once again, just your standard GLM matrix factorization. So some of us might have seen this in other contexts. So just imagine I have my original raw single cell count matrix. So this is the matrix where 70+% of the values are 0. For those of us who work with count matrix, it's very, very common to use a negative binomial. You could just imagine negative binomial is a slightly sparser version of a Poisson distribution. It's a little bit more over dispersed, but nothing too special. It's a negative binomial distribution with some mean. And this mean is going to come from some terms where I have some covariates for each cell. So once again, for this cell, this might be the age, sex, smoking status of that donor that contributed this cell. So that's going to be an observed matrix. I observe my count matrix. I observe this covariate matrix. And what I'm trying to estimate is essentially the coefficients to these covariates plus some latent factors. So this is usually what matrix factorization GLM works. So I want to then estimate my residuals in some sense as a latent matrix itself. And this is how I'm going to pull together a lot of information across different genes. And so once again, X and Y are going to denote, X is going to be the latent vector for each cell, Y is going to be a latent vector for each gene. And this is what helps me pull together information across all the genes, across all the cells, so I can help bolster the signal in my very, very sparsely observed genes. And so in your negative binomial, you also have an over dispersion parameter. For those of us who've been using negative binomials before, this is just a parameter you have to estimate so you get a better sense of what the variance is. There's some more substantial details, but I'm going to just brush them under the rug right now. So this is basically the model. You can imagine I'm going to say just a couple more slides because there may be some people in the audience who works on matrix factorizations. So basically, in a matrix factorization, what has become somewhat well known, at least from the theoretical side, is that the initialization matters a lot more than your updates, because your updates usually can just be gradient descent. You're going to be in a good spot as long as you had a reasonable initialization. So a lot of the statistical theory work have focused more on how do you initialize properly, and then the updates are pretty standard. If you initialize well, you're going to end up in a good spot. This is going to be a non-convex problem because this optimization, if I just broke down the negative log likelihood of this model, is going to be a non-convex function, which is why I have to worry about the initialization before I start doing my gradient descents. So in the first part, I just regress all my observed covariates C onto other way. I'm going to regress each gene in the count matrix A onto my covariate C. This is just a really fancy way to explicitly write down what the GLM looks like. But this is just a GLM, just take my word for it. And then I'm going to compute some residuals based on the log of the count matrix. And then once I have the residuals, I'm going to decompose it. I'm going to initialize it to either my embeddings of the cells or the embeddings of the genes. So there's nothing that impressive about this, but it's more impressiveness in the engineering details. The high-level details are pretty straightforward. First, I just do a GLM, and then I take the residuals and plug it into SVD. Then, as I said before, in this literature, it seems like the initialization matters a lot more than how you update it. And so the updating is not going to be that fancy. It's just going to be an alternating minimization. Now that I've initialized it, I'm just going to do an alternating gradient descent. I'm going to either optimize the cell embeddings or optimize the gene embeddings and just alternate between these two. once you fix one latent embedding, the other one is just a convex problem. So we can solve it exactly. Things of this nature. I'm going to add some penalties everywhere just to make sure that I regularize and don't have any bizarre like latent embedding. So you can imagine these are more like ridge-like penalties. So there's some clever engineering details, but I would say overall, this is a pretty standard method. So now that I've estimated my latent embedding, yes.
So often those problems are underdetermined,
Mhm.
and so there's an identifiability problem.
Mhm.
There's many local optima.
Yes.
Do you have that difficulty, and how do you address it?
So there's two answers. One is more satisfactory than the other, but both are true. So identifiability in a sense that given a solution, given, let's say, one bit of, let's say, so the literal but not very useful answer, which I'll say first, is if I ask what is a fit that has the same log likelihood, I can identify the unique parameterization that gives you this likelihood. That's not a very useful answer because your question was really about the different local minimums. Right. So my first answer was given a local minimum, I have a unique parameterization of it. Not very useful, but yes, technically true. The part that is not as satisfying, which I will admit, I haven't seen many papers to satisfactory overcome it, is that we're stuck in a local minimum in some sense. So that's why in a lot of these non-convex optimization papers, it seems like your initialization basically dictates the performance of the entire method. It's very hard in these type of procedures to jump from one basin of traction to another.
Right. So one way to deal with that is multiple starts.
Yes. So there's some engineering tricks we can deploy, which we do in our software. But in terms of like a satisfactory one yeah, so with multiple starts, I can't really guarantee that I'm gonna ever find the best. So yeah, there are engineering tricks, but in a conceptual sense, I would say it's still not clear. Given a fit, how do I even know if I'm in a best local optimum or the worst local optimum? That one I think is still an open question.
Right. Okay. But one thing you can do is ask, if I do S starts versus 2S, 4S, do I see a better local minimum after more starts? That gives you some. It gives you like some clues. Yes, it gives you some notion of how many starts you need for a particular problem to get to a good local. At least empirical.
Yes. The other way, might as well say it because it might be of interest to the crowd. The other thing I kind of magic trick like disappeared is the choice of K. Now there's some literature to suggest that one other way to test if you have a global optimum is if I fit it for K and then just I initialize at 0, the K plus 1, I just manually in the middle of the method, I just add the K plus 1, I set it all to 0, so it technically doesn't change anything. And if I optimize starting from the fit of K, I add the K plus 1 initialize 0, and it doesn't change much, then there's some theoretical evidence that means that I have the global optimum also. Now, I haven't had a student to try this wacky idea yet, but I am slightly curious about this. So there's some creative statistical ways that I think could be used. I just don't know if they actually work yet. So that's another way to test. You basically change the value of K and ask if my K plus 1 variable changed at all. Another creative way that I think might be of interest to some of the students in the crowd, but we haven't gone around to trying it yet. But after I do this denoising, after I do this imputation, as I said, I want to overcome this type 1 inflation, and that's all going to be on this slide, which I'll just summarize. So you can imagine this Uij is this manifold, quote unquote. I just take my fitted low-dimensional embeddings of my cells and genes and add it to my coefficients and covariates. Because I'm working with negative binomials, the length function is going to be my exponential function. And what I'm going to do then is I'm going to weigh it against the observed count and the fitted library size for that cell. So essentially what this is going to do is if you worked with these posterior distribution before, you'll realize that on the left-hand side of the denominator or the numerator, this is just my denoise gene expression. On the right-hand side, I'm weighing it against the actual data itself. So if the data itself was very, very sparse, I would have no weight over here. If the data was very, very dense, then I would put more weight over here. And that's all scaled by my estimated library size. And that's how I'm going to kind of tamper to make sure my type 1 error doesn't go through the roof. And so we can assess if a gene is super, super overly dispersed. I probably should not trust it. So I probably should trust the original counts more than my fitted ones if the gene is very, very over dispersed, and so on and so forth. So I can answer more questions about the literal details in just a little bit. So, okay, so back to my results. So now that I told you a little bit about how our method works, so you might ask, why did I do this lung analysis on T cells? And at the time, the main reason was because we had two biological replicates of this. So two completely different set of authors studied the same disease, completely different people. And our premise was if our method was worth its weight, then I should find very similar findings in both cohorts of people studying the same pulmonary fibrosis disease, focusing on the same cell type. And so you can imagine here, these are just volcano plots, either from the Adams data set or the Haberman data set. This orange line denotes the threshold for multiple testing correction. What's shown right here is all the green genes are quote unquote housekeeping genes. These are genes that are just essential for basic survival for the cell. So usually these are treated as negative controls in some sense. You don't expect the disease to kill the cell because if it killed the cell, you wouldn't see it. So usually we don't want to see any of the differential genes as these negative control housekeeping genes. That's more of like it's not a hard rule, but that's a guiding principle that we like to use. And then I can also mark which genes the authors originally found or the genes that my method found in orange. And so what am I going to do now? So now I'm just going to take all these genes and correlate them. And that's what this hexagon plot shows. So basically in each grid of this hexagon, I'm going to have a color bar of how many genes landed in either the Adams analysis or Haberman analysis. And I see some reasonable correlation. This isn't super high, but given that these are completely different cohorts of people, completely different technologies, we are pretty happy with the fact that they were at least somewhat correlated. Now, if I used a pseudo-bulk analysis, DEC2, you'll notice that the correlation goes away because a lot of your power just vanished when you collapse all the donors to just one cell. So these are some of the reasons why we chose to focus and showcase this particular analysis was just because we had two completely different data sets that could serve as biological replicates. Now, ever since we finished this paper, there's a lot more studies that we could counterbalance and try a different thought experiment of, and I could show you some of that in just a little bit. But that's the reason. And then in the last section, just so you can see a little bit more. So we also did this for an autism data set, which at the time was one of the biggest cohorts we could find for studying autism. And this is, once again, my volcano plot where, once again, I'm marking the genes that I'm using as quote-unquote negative controls in green, your housekeeping genes. And I can compare it against a pseudo-bulk method where I can see that, there's more meaningful genes that my method found, and this is just the hand-wavy way to say our method seems to be revealing some interesting signal. I'm going to show you a little bit more convincing evidence in just a little bit than what we originally showed in the paper. But suffice to say, that was what we did. So what's next? So before I move on, I just want to say that for some of us who work on genetics, this whole talk might seem not very useful because in our SNP analysis and our GWAS, we are constantly imputing the SNPs because different consortiums, different labs might use slightly different chip arrays, genotyping arrays. So, you basically in a GWAS analysis, you then upload your SNPs that you measured on your array to some imputation server, like Michigan's, and then you basically get all the SNPs and all the possible locations, and then that's how you concatenate your data sets across different consortiums. So in the GWAS world, we're constantly imputing and then doing differential expression testing, and that has never seemed to bother the geneticists who work on these massive 10,000, 100,000 people analyses. And you might wonder why this story of imputation inflating type 1 error just doesn't play a role in genetics. And so here are some reasons that I think this phenomenon is. I don't have any evidence of this, but this is when I think about this question because I was asked this. So certainly in SNPs, you have a large linkage disequilibrium. So SNPs in one location are highly predictive of SNPs in another location. So that's one reason why just imputing is just a lot easier in genetics than in gene expression. But also there's usually when you design your cohort, you assume that the population structure in your cases and controls are balanced, which is also not quite true in a gene expression data set. And then the last one is, yeah, there's just so many SNPs, but each chip can only measure so many. So there's also practical necessity, all of which are slightly different in the gene expression world. Happy to talk about this because
there's another simpler answer, which is there's just 3 possible genotype values that I just
Oh, yes. That's probably a good one I should add to this list.
For error.
Yes. So I should probably add that one to the list. So these are some of the reasons why on the genetic side, this problem doesn't seem to be a big deal, but on the genomic side, it seems to be a disaster. So let me just say with the last 5 minutes, some of the work that I'm doing now that this paper is done. So this slide is all to say that I've been pivoting the lab to work on more Alzheimer's disease related things where in particular, we're interested in how the microglia plays a role in this Alzheimer's disease. So as a one sentence summary of both parts of this picture. So Alzheimer's is characterized as when you're elderly, think 65 or older, your memory starts to decline. And one of the findings over the last decade that has been promising is that a lot of the SNPs that people are finding implicating Alzheimer's happen to be genes that seems to be more expressed in microglia. So this is why there's a slightly disproportionate interest in microglia among all your cell types. There's probably more interest in microglia than the other brain cells for studying Alzheimer's. It's just because of this reason. There seems to be more evidence that microglia play a disproportionate role. So given this, we're trying to understand, let's collect as many people as we can get our hands on and try to figure out what's differentially expressed between the microglia in people who are diagnosed with AD at time of death versus people who have good brain pathology. They don't seem to have AD. And so once again, we're going to return to this example, but now in this follow-up work, which isn't about eSVD, it's a more general work now. You can imagine this is a slightly tricky question now, because when I think about this experimental design, this is something my collaborator asked me, and I have no answer to it. So if you have any suggestions, I'm very open to it. You can imagine as the person running the sequencing, I might ask, given my limited budget, should I sequence more people or should I sequence more cells per person? And this is like a power calculation that does not exist currently. And so this is the question that we're going to try to study using our methods. Given the constraints that I have only so much money when I'm designing my panels of donors that I should collect and get brain tissue of. Should I sequence my precious donors more deeply, or should I try recruiting even more donors? And what's a trade-off? Is there a sweet spot somewhere in the middle? And so this is where we took basically, this is like 3 out of maybe, to my knowledge, like 5 data sets that are of large enough quantity of just microglia. So we have 3 data sets. We're just looking at the microglia population in these data sets. What these bars are showing is that these data sets have slightly different qualities. They have slightly different number of nuclei. So in a simple sense, this first data set has the least number of people, but the most number of nuclei and the most number of nuclei per donor. And on the other extreme, this data set right here has the most number of donors, almost 10 times more, 340 people compared to 22, but it has very, very few nuclei per donor, only 100 cells per person of these 300. Whereas on the other side, you have 22 people, but you have 5,000 cells. So we have a range of data sets. And given this, we have some hypothesis. If I had different methods and I deploy them on this, I should expect, something. So this is what we're going to study. Using these 3 data sets, we're just going to use it as a playground to test our different ideas. This slide is just to show that there's some covariates we're going to adjust for. In Alzheimer's, the most important covariates to adjust for is this specific genetic mutation. If we study Alzheimer, I will tell you a little bit more about this, but suffice to say, there's some covariates we're going to adjust for. And all these data sets are basically as biologically close to one another as possible. They're all from microglia, all from the prefrontal cortex part of the brain, and all from frozen tissue. So we're trying to make it as comparable across the 3 data sets as possible. We even adjust for the same exact covariates in each of the analysis. And now, let me just tell you in the last 2 minutes what we are going to do. So instead of asking the more power calculation related question, let me just ask a simpler question, which I can just experimentally see if it's true or false. If I recruit more donors, does it really matter which differential expression method I use? So say I had 3 differential expression methods. They all are reasonable. You might imagine kind of like in the sense of law of large numbers, even if I have different estimates, if my cohort size was like infinity, maybe as long as I started with 3 reasonable estimators, they start converging more and more to one another. Because you can imagine, even though there's technically many ways to do differential expression analysis, maybe if my cohort size is big enough, this distinction between different methods isn't as important anymore. Because maybe the difference between these 3 methods is simply a matter of power. But if I have enough donors, it doesn't matter how low my power is, I will eventually find the same things. And so this is what we can start doing. So with my master's student, we're going to literally run 3 analyses on each data set and compare the results between different methods. So the way to read this plot is that the x-axis and y-axis are going to be the results of 2 different methods on one data set. This is the sine negative log 10 p-value. So it's just a negative log 10 p-value with some information about the sign of a log fold change. And that's going to be the x and y-axis. And I'm going to mark in purple the genes that are differentially expressed by both methods, because one method is going to be the y-axis, that's your mixed effect model method in pink, and then one's going to be my blue pseudo-bulking analysis. And I'm going to also record how many differential expressed genes are in the intersection between these 2 methods or uniquely found by one method but not the other, or all the other combinations, my union. And so this is for one data set comparing 2 methods. I have 3 data sets and 3 methods. So you can imagine I can do all 3 choose 2 combinations on my one data set. So you can see that these 2 methods, a mixed effect model method is pretty correlated with the pseudo-bulk method, that's 0.9, and finds a reasonable overlap. My pseudo-bulking method and the method I just told you about in purple, a little bit less correlation, a little bit less correlation here. So this is on one data set. I can also fix the 2 methods I'm going to compare against, a pseudo-bulking method and a mixed effect model method, and just try it on all 3 data sets. And we'll see that the correlation is usually pretty high. So this is just say that in this particular case, a mixed effect model doesn't really buy you much compared to a pseudo-bulking. That's more of a property of this particular system that we're studying. Even across 3 completely different data sets, different trial designs, different sequencing strategies, different consortium sequencing it. And now, of course, I can do this on the rest. I mean, we're not going to stare at this. There's a lot to take in at one time. But let me point you to the thing that we found curious, and then I'll call it a day. So you'll notice, focusing on this middle column, this first column had only 22 donors. The correlation between these 2 methods is 0.5. When I move to this middle column, this is over 80 people. The correlation goes up. These 2 methods are agreeing more and more. But now when I move to 300 donors, the correlation is not nearly as high as with a different set of 80 donors. So this is particularly curious to us because my whole premise of why I had my master's student do this is I thought that if I had 3 sensible methods, as my number of donors increased, my method should agree more and more, because I thought the only difference between these 3 methods is an issue of power. But that wouldn't quite explain why the correlation then drops if donors was the biggest factor that I thought was going to drive the power. And so this is what we're still working on, and I'll love to hear any thoughts. So we might have some hypotheses on why on earth this is phenomenon happening. The first one is maybe it's not just the number of donors that's driving the power, but maybe, in fact, the cells per donor, that number is also driving the power, because in this large data set with 345 people, I had by far the least number of cells per donor, compared to this data set, which had the highest correlation, not as many donors, but certainly more cells per donor, so maybe that matters. Who knows? The other one is I kind of slid under the rug that I pretended all the microglia are the same, but certainly we have different types of microglia, which I am not accounting for. I'm just doing all the microglia from any case donor versus all the microglia from a control donor. And so maybe my analysis is confounded by the microglia subtypes. Maybe that's another hypothesis. And the third one is, well, there's no reason this premise had to be true because I have a pseudo-bulking method, I have a mixed effect model method, I have a matrix factorization method. The statistical targets for each of these methods could be actually quite different. So there's no reason as n goes to infinity, they actually converge. So who knows? So we're still working out. We have some analyses planned to rule out yes or no. Is this the reason? Is this the reason? Is this the reason? So that's what we're wrapping up to finish this particular benchmarking exploratory work. Yes.
So having worked a bit with the ROSMAP data set, I can suggest a hypothesis which literally but not technically contradicts your assertion about batch effects. So I think part of what's going on in the single cell world is that quality control has gotten much better as time has passed. So what's wrong with the ROSMAP data set is it's old.
Oh, yes.
That's not fixable.
And
so what you're getting is that with newer data sets, the lab work was just better.
I see.
And that's what we're finding in our single cell work, is that the newer data sets are better just because people learn from their mistakes.
I have to think about that because certainly we're looking at one particular brain region that is one of the older ones, but then ROSMAP is supposedly releasing a different brain region sometime soon. And I wonder.
So you can test the hypothesis that the newer data should be better.
Yeah. I think that'll be incredibly fascinating. Yeah, I haven't thought too much about if batch effect is driving. Okay, but we'll see.
So it's not the usual meaning of batch effect.
Yeah.
Right? Because it's not about the same lab was doing these experiments. It's more generally that the field is advancing, and it's not just the statistical methods, but the lab work is advancing too, and people are doing better quality.
Yeah, we'll have to see. I'll be very excited to see. Yes. But yeah, just to wrap up. So in general, this work was about our single cell method to test for differences on the cohort level, where the main novelty was to account for the fact that we need to adjust our denoise single cell data. We used the dimension reduction framework to do that. And that was so we can remove the strong covariate effects while still sensibly testing for differential expression. There's a bunch of things that I would encourage you to ask me or just read the paper to figure out the design of the test statistic. I definitely swept that under the rug. I said a little bit about how to handle the non-convex optimization, but certainly I didn't even tell you about the tuning parameters, the diagnostics. But the main takeaway along the whole is that outside of the genetic field where imputation seems to be sensible before you do multiple testing, in most fields, it's to my awareness that pulling information is sometimes a necessity when you want to remove for strong confounding effects. But you don't have to use our solution for it because the context in a different system, different type of data might be different. But certainly this is just to say that something should be done after you denoise to account for the fact that you denoise before you do a differential expression testing framework. And thank you so much. So this is just my master's student who helped me do a lot of the stuff in the last half of the talk. And with that, thank you so much.
Question about the we can also consider an active model. So do you mean like the main difference between your method and your method? We're applying for the post-doctoral like the level population where it's like more similar.
I would say so. So I would say compared to let me exit out of so I would say compared to DE-seq2, the main difference outside of the literal test statistic is the fact that I'm going to first do this matrix factorization and then do the testing part. So I would say conceptually, there's just these two differences. My test statistic is slightly different because I want to count for donors, but the other one is I did this matrix factorization in the middle. But otherwise, outside of like literal engineering details, it's conceptually very similar.
So several questions about the visual contrast and we can follow up later, but maybe one question is there an evaluation framework to validate your denoise data that can keep the biological signal where it introduces bias?
Outside, this is a fascinating question because this is the only reason that we did this, because currently I don't have a better answer. So currently it's not really validation because I mean in some sense it's validation. I would say it's like kind of validation. I wouldn't say this is the most convincing validation ever, but yeah, so far when I try these methods, it's more just like if I have two completely different data sets, do they find the same thing? This is not the most convincing validation. I would say more convincing validations will probably be some follow-up experiments, but it's a little bit tricky, especially in the AD scenario that I'm working with, because you can't easily perturb brain cells. You can do it in cell lines, but then my analysis was done on humans, and then I wouldn't naturally expect the findings in cell lines to always recapitulate the ones from humans. So it's a little bit tricky. I don't have a great answer of this outside of the usual like computational trick. So we can look at the pathways, make sure the pathways make sense. But I found so far the only like way that I know how to do, and I also think it at least carries some water, not the most water, but some water, is just to find like biological replicates. But I'm always interested to hear more suggestions because I'm not super happy with it. I wish I had another method, strategy in my back pocket, but I don't currently have one.
Another question about pseudo bulking. Does it matter? So let's say you bulk an individual, all the cells in an individual into one row in your matrix. Does it matter if you pseudo bulk by sort of bootstrapping across patients?
Ooh.
To make it would be a way of getting it with patient number versus cell number issue.
That
Be like a bootstrap randomization. So if you pseudo bulk across patients, you would actually serve as a randomization of the patient while assessing whether you're actually sampling the same type of cell in different individuals.
I've never thought about that.
Okay, we'll write a paper.
Yeah.
Because that would actually orthogonalize the patient effect in the cell, the zero cell matrix.
I don't need to think about this. Yeah.
It's like bootstrap analysis in phylogenetics. Randomizing.
Oh.
It's not like subsetting it. It's just reparsing it by going.
Oh. I have to think about it. That's a very curious idea, though.
So one difference from phylogenetics is here you have to account for the fact that there's different numbers of cells sampled from the different donors. You can try to normalize that by subsampling, but then that leads to another dimension of randomization.
Yeah, you can trim the microbial diversity, trim down to the lowest common denominator of all cells, and then pseudo bulk on those or random. Yeah, it'd just be interesting to try it to see if you end up with a result halfway between your two extremes.
So I think the main thing I'll just need to make sure I think about is when I randomize the cells from donors, I need to figure out. So the nice part about pseudobulking on the real data set is all the cells from the same person has the same human covariate.
Have the covariate. So
and then now when I shuffle this, like if this cell moved over here, I guess I could just keep the gene expression, but now give it someone else's covariate.
I suppose you could pseudo bulk among individuals with the same covariate.
Oh yeah, I mean, yeah,
Certainly, maybe pseudo bulk randomly without respect to the covariates. It would potentially randomize the effect of the covariates.
yes. Well, I'll have to think about this.
Somebody was called.
Yes. Well, thank you for the suggestion. All right.
Why don't we just get better data? 70% of the matrix is.
I mean, it's across many cells, so
So
You had better data, wouldn't you?
yes. If we had if everyone had a multi-million-dollar experiment, if everyone could get data set like this, I think a lot of problems would go away. But yeah, it's we're at the mercy of how much budget.
On one hand, the better data are coming, but the short answer to your question is, for cost reasons, 10x and engineering reasons, 10x technology, which has a lot of technical deficiencies, won out over SmartSeq 2 technology, which would give you much
Yes.
better data. So we have found in some scenarios that basically you can only get a reasonable answer to SmartSeq 2. 10x is useless. So for example, 10x gives you only the 5' aggregate with 3'. So if you want to do something like false variance,
You're missing a lot.
it doesn't work. You have to use SmartSeq 2, which gives you the full.
So right now you're focusing on a single cell type.
Yes. So all of these analyses, even in all these categories, usually you first identify the cell types and you say, I have a pure set of cells, and then I only yes.
But now that you're working at the single level, you see.
I have seen some work on this. I think it just depends on how much I believe, let's say, like my signatures in microglia will be recapitulated in neurons.
Right, but when you're trying to compare now,
Mm.
you're still putting across the defined cell type.
Mm.
Finding it. So are you like as part of the idea should be that you go to one cell on one people. That's
Mm.
right. And are you using that signal, not just people that the initial definition of the cell groups.
Potentially. I think the whole field has not quite decided what cell type actually means. Right now, by cell type, I just mean it's the result of some clustering method based on gene expression, which is not very satisfactory. Well, thank you.
Thanks for coming again.